Okay. So welcome to this workshop series. As I said, it's quite a specific niche workshop. We are interested in how to integrate multi-omic data into human exposome study. And really here, the environmental factors part, we think it's quite a novel piece added to the multi-omic data integration and requires specific data integration and thought. So today is the first session, and actually the time of today is a bit more than an hour. It will be an hour and a half. So it's I'm giving a 30-minute introduction, and then there will be an hour talk by Arthur. The next session will be only an hour, weekly on Wednesdays at the same time, except next week because it's a Catalan bank holiday. So please have a look in the website. I actually show you here. I'm sorry, with Zoom. I can't access it. But if you go on the website of the workshop, there is a PDF with a detailed program that we will keep updating, since some of the session later on actually have 2 speakers and we are still refining the program. So the introduction to this workshop, I will start, and then it will be followed by Augusto Anguita, which is a postdoctoral researcher in our group. Since in this group, there are people that are just becoming familiarized with exposome concept, I'm just giving a quick introduction about exposome and multi-omic. And then the rest of the talks will be actually quite advanced on the data integration and won't touch more on the basic concept. So so here in the exposome concept, it's really the integration of the external and the personal exposome, which are measured through different tools at different levels. And here we are interested in how they are associated with biological responses, which are measured through omics markers. So what do we understand with the internal exposome? It can be the individual physiologic and biological responses to environmental factors needed for obtaining homeostasis, and this has links to many disciplines. So this goes beyond environmental epidemiology. We are working with molecular biologists, with toxicologists, analytical chemists. I'm sorry, I'm admitting people while I'm speaking. Okay, and what do we mean with new omics tools? Here we are thinking of any omics that are post-genomic era. So with the genome, we've been able to sequence the DNA, but now we are also interested in more the gene transcription, the epigenetic marks, the proteome, the protein markers, and the metabolome. These are the main omics tools. And I will also touch upon the microbiome, which is not here. All these omics have I use different technologies and quantify different pools of biological molecules, but that's the main characteristic of all of them. We're not focusing on one single markers, but on a holistic characterization of biological layers. And this is the integration into exposome studies is linked to key advancements in technology. And here I'm showing some milestone in different omics analysis. You can see from the 60s where we started to be able to decipher the genetic code. It took a while until we could actually analyze the transcriptomic, actually in 2010. The proteome, it's only in the past few years, I would say, we can pretend to be able to use mass spectrometry development to sequence more than 100 proteins. And also there has been a big push towards exposome and metabolomic analysis. through also development of liquid chromatography, gas chromatography, and mass spectrometry with much more accuracy, higher resolution, which allows to measure a much larger dynamic range of molecules. So we can now pretend through one assay to measure both endogenous biological molecules and external molecules in the same chromatography mass spectrometry assay. There's been also great advancements in lipidomics, which are very complex chemical compound to characterize due to their similar weight and structure. And also, finally, there's been advancement in wearables, which are very important to characterize the external exposome, but in a personal way. For example, through the development of air pump and sensors, which are now able to profile both biological and chemical exposures. Sorry, I'm just trying to hide. So some of the main frontiers in the measurement of the internal exposome has been the development of technologies from small to high throughput to be able to screen omics technologies and exposome. We need a large sample size, such as in genomic studies, but this has been hindered by the cost of omics analysis. But as you can see, for example, for proteome analysis, the price per sample has dropped from $3,000 to $300 in less than 10 years. And this is the case for all omics analysis, which allow the screening of much more samples and gives us the right sample size to start to do this kind of analysis. We've also moved from analyzing knowns to unknowns, from targeted measurements of a few proteins, for example, 100 proteins to 1,000 proteins, and also able to measure unknown proteins or chemicals in omics assay. So we have seen an evolution in research from hypothesis-driven investigation to discovery-based exploration. And as I said before, I think the top technology that have pushed this field has been the development in mass spectrometry, high-resolution mass spectrometry, and next-generation sequencing, in particularly important for microbiome metagenomic analysis. And finally, which is of interest for us now to talk about data integration, there's been a great improvement in computational integration with development of tools for dimensionality reduction. We'll speak today about mediation analysis, predictive modeling with machine learning, which are enhancing our capacity to integrate diverse data sets. So what's the motivation behind integrating multi-omics? Well, human disease or preclinical assessment is complex. And if we use the elephant and blind man parable, with only one omic, you're actually focusing on only one part of the biological system. And all omics have their shortcomings. Even epigenetic, which has seen much better advancement and gene annotation and functional annotation, still, actually, with the current arrays, we only measure a small part of the genome through epigenomics. So you combining different omics technology, it's also complement each other and the shortcoming of one another. So here I'm going to structure the rest of my introduction around these three blocks of premises of why do we integrate omics in exposome studies. This is a non-exhaustive list, but I think these are the main objectives of integrating these tools. And the first one is to better understand biological mechanism. And that's very important into understanding environmental risk factor. Because when we link environmental factors, one of the aim is to, for example, influence policy. But to do this, we need to prove the biological plausibility of this association. The second main objective is to predict past exposures. For example, predicting from pregnancy. We know that early life has a strong influence on the rest of disease progression throughout lifetime. But usually, for example, if we design a clinical study and recruit patients in their adulthood, how can we measure back their past exposure? Not only based on the questionnaire having recall bias, but using objective measurements through omics measurement. And the third objective, we aim to predict future health outcomes before the clinical symptoms appear. Here, the main focus is prevention of disease. So through omics, can we detect early markers of disease and the impact of exposures on it? So this is very much linked to the shift of paradigm in medicine to personalized medicine from treating disease to prevention. And the first point, I already talked about the prevention and the personalized medicine. The point is also to be able to intervene, and maybe by better understanding the molecular effects of external environmental exposure, maybe we can find mechanisms to which we can reverse this association through drug, diet, lifestyle change, or biotic intervention. Here, just a quick reminder of what can we call a biomarker. Biomarker, there is a bit of mix of terminology because we come from different disciplines. I found it important to remember that a biomarker is a measurable internal indicator of a molecular or cellular processes, but it's quite a broad meaning. It can be biomarkers of exposure. For example, when we measure pesticides, metabolites in urine samples, this is closer to the exposure. We can be markers of effect, more related to the biological response, for example, inflammatory proteins, cytokines. And biomarkers can be around the disease progression path. They can be early predisposition biomarkers, prognostic biomarkers, or diagnostic. Here with Exposome, we are more interested in early biomarkers. Okay, so just a sort of the first point, the first aim of including omics on how to the aim to understand biological mechanism. Why do we want to understand this? Well, it's important to have some basic scientific knowledge of the toxicological effect or to understand common biological pathway of action of different pollutants. So if they have common targets, and also for providing evidence for public health policies. And how can the exposome affect biological processes? Well, through alteration of the genome, but also mainly through dysfunction of biological pathways. And just to note that when we talk about biological pathway, actually, it's quite a complex process. For inflammation is often mentioned as the main biological pathway affected by environmental exposure. And that involves many types of molecules, from proteins, from bacterial signaling, from cell counts. So this is just to show you the complexity of how to measure the dysfunction of biological pathway, providing more evidence that we need multi-omic layer to measure this. If you don't know this review by Anna Peters and colleagues, I invite you to check this. So they came up with what are the main biological signatures of environmental insults. It's a non-exhaustive list, but it does provide food for thought of what are the main biological pathway of interest when we're looking at exposome. I'm going to skip through some of that, but I will share the slides. So what the second objective is to predict past exposure. And why would we want that? To allow us to identify exposed individuals more accurately. Because, for example, if we're only able to measure acute exposure, that's limiting the analysis of accumulated exposures throughout life course. And also to anticipate potential future disease linked to the exposure. And what characteristic do we expect biomarkers or omic marker of past exposure, where this should be a persistent signature? And this really has been more demonstrated with epigenetics up to now, which is a good marker of cell memory. We also, as a main characteristic, expect a dose response. For example, here I'm showing an example of epigenetic markers for prenatal tobacco smoke exposure, and we can see a clear dose response in the Helix study between different level of tobacco exposure, from second-hand smoking exposure to sustained exposure to direct active exposure. And there's great examples now on how to create such predictive score of biomarkers. And for this, we don't need causal relationship. So this is a difference with the first objective of omics, to understand biological mechanism. Here we want causality, but in this second point, to just predict past exposure, we don't need strong biological understanding. And we're going to show several statistical methods to do this, from linear regression-based model to machine learning. And finally, the third aim is to predict future health outcomes. That's the aim is to predict disease risk, but also disease progression if we are recruiting people with already an existing clinical manifestation, or also to analyze the response to treatment. And also there's been a lot of research on looking at accelerated aging. And I'm going to give a quick example here with the development of epigenetic clocks. In this case, we don't have, as I said, any a priori hypothesis. We just use the whole epigenetic signatures to predict the biological age. And there's been also further development using even metabolomic clock. This can then be used to analyze the association with external environment, such as in the Helix study here, where we found that in children, air pollution, toxic chemicals, and smoking were associated with accelerated biological aging, even in childhood. I would like now to speak a little bit more about the challenges of including omics in exposome studies. And to do this, I'm going to take this scoping review we've done on endocrine disruptors studies in humans that employed omics. And we had quite interesting outcomes. So we have actually screened 100 studies that used omics, and we looked at what were the main chemical analyzed in omics studies, and actually to our surprise, they were still very focused on single families of chemicals. And actually most of the studies focused on still targeted approaches. So they had a hypothesis driven, focusing only on oxidative stress, or only on lipids and insulin, or steroid hormones. But now through multi-omics, we can actually screen all these known biological pathways and discover new ones. And some of the study design problems we identified is that most studies lack power to detect subtle effects of endocrine disruptors, as it's expected in general population. And this effect is even more likely to be diluted in a small sample size. And we expect a high exposure misclassification due to endocrine disruptors' intra-individual variability. So this is a one example of a type of exposure, but it can be also valuable for other type of exposure. So I would like to summarize here the challenges of multi-omic integration in exposome studies. We have a problem of small association effect. Omic studies actually have been initially developed in cancer studies, clinical studies, or occupational studies. But now most of the exposome studies are in the general population. So we expect subtle effect. And that's where we need to think what kind of statistics are necessary to do that. And this small association require large data sets. Another challenge is that we're interested in exposure mixtures, interaction. So our predictor of omics alteration is actually multiple, and it creates challenges into modeling these cocktail effects. We're also interested in mediation analysis, which becomes also complicated when we start to have thousands of mediators and hundreds of exposures. Another type of challenges is the biological interpretation. Even if we are measuring very well a gene transcript level or a metabolite level, if that omic feature is not clearly linked to a functional effect, it's going to be very difficult to give a context. And we found in our previous studies that most of the omics markers have been, we're using, catalogs or, for example, KEB pathway or disease catalog of this omics for functional analysis. But most of the information comes from the context of cancer research. So when we start to translate that to children, it's not always relevant because biological disruption in children might not be related to cancer, but earlier markers of phenotypes. One other point is that omics vary across time within individuals as well as exposure, and not all omics markers will be relevant to characterize long-term exposure. This is where it's important to check into panel studies or repeated sampling and really understand what are the omics marker we find. I'm giving here the reference of a study where we measured at 2 time points multi-omics analysis, and we characterize which omics are stable or not across different seasons. Another challenge is that even if we are using several omics, they can still be confounded with the shared determinants, for example, BMI or sample handling. And by combining the omics, you're increasing even more the confounding structure. Also, there is an importance of thinking where are we sampling and where we take the omics. For example, typically epigenetic markers, they are measured in white blood cells from blood collection. And here we are measuring only immune cells. So that's just something to keep in mind. We're not necessarily measuring the omics in the target tissue. And our omics also, as I said, there is a strong component of time. Omics readout provide a snapshot of the biological response. And we need to think when we look at our exposure, which time scale are we interested in? And I show this review where we can think of the time scale is very large, from transgenerational time frame to very acute to minute to hours of changes in omics. Okay. And now I'm going to give the floor to Augusto, who is going to talk a little bit more about which framework, philosophical framework, we can use to combine different multi-omic datasets.
Hello, can you hear me well?
Yes.
All right. So thank you very much, Léa, for this introduction. So the idea now is to give a brief overview of the different approaches, statistical approaches that we are going to cover in this workshop to overcome all these challenges that Léa has already presented for multi-omics integration. Fortunately, in the past years, different advanced statistical frameworks have disseminated with different statistical solutions for these problems, like dimensionality reduction or feature selection. That is also a big issue, especially given this problem of the sample size. And I think it's important to have a kind of slide about the taxonomy of these methods, also to give you a good idea of which session will cover which, and you can decide better also which is of most interest to you. So regardless of the solution that these methods have or provide to overcome these integration challenges, depending on the experimental situation that we face, we can have different type of approaches. They are divided in horizontal integration approaches or vertical integration approaches. The horizontal integration methods, they face the situation where we have multiple omics layers belonging to the same type of omics. So it will be the same nature, like, for example, gene expression, but we have multiple conditions that it could be different time points for the same individuals or different populations, and we want to assess the different effects across the different populations. And on the other hand, we have the vertical integration approaches, which involve the analysis of different type of omics in different layers. I think, Léa, you quit the presentation mode. Okay, all right. And this is what we have here in the bottom part of the slide, where we have also a second division of the vertical integration approaches. We can consider hierarchical integration or parallel integration. And the main difference is that in hierarchical methods, we can incorporate a kind of a priori knowledge to guide the integration process. At the time of extracting latent factors, for example, we can specify if a specific omic should have a more pronounced or higher impact on the health outcomes, or we can even decide which type of correlation do we expect among the different omic layers. Or in parallel approaches is those cases in which we don't have any a priori hypothesis and we just integrate the different omics at the same level. So it's basically in the vertical integration methods in which we are going to focus in our course. Next slide, please. And here we can find a second strata division where we have early integration method, intermediate integration method, or late integration method, depending on how do we deal with the different layers of data. The basic one is the early integration method, where we have, let's imagine that we have 3 matrices of data, and we just concatenate them all, and we form, and we end up with a single matrix with all the heterogeneous data, and we will apply, after proper normalization and standardization of all this data, a single model to extract either predictions or cluster. Doesn't matter if our purpose is prediction or is an unsupervised approach. On the other hand, we have these intermediate integration approaches where we try to account for the particularities of each omic layer, and we try to build a specific model for each. But these are not completely independent models. Models built for each omic layer probably will share parameters during the estimation process, and this will make an integrated model, either supervised or unsupervised, for the final output. And finally, we have the late integration method, where we indeed build totally independent models for each omic layer. This sometimes is motivated in the particularity of some specific data source, and we will finally combine the output of these models, either doing meta-analysis or for majority of voting in case we are facing a prediction task. Next slide, please. So this is kind of a summary of the whole workshop structure. As I said, most of the methods that we are going to present lay into the vertical integration approaches. Some of them, they will present early integration approaches. Others mainly will present intermediate integration approaches. But here is the overview. We have tried to cover different topics that we think are important, especially in the context of exposomy, where the mediation role of omics for environmental exposure is the main thing that we want to look at it. And we will have these 5 different topics. The first one is dimensionality reduction, is the one which we start this session. Dr. Arthur Tenenhaus will present an overview of different dimensionality reduction methods, presenting the basic concepts, also the different type of methods that can deal with either 2 layers of data or more layers of data and some particularities. This kind of introductory session to dimensionality reduction that is basically the most often used approach for integration. Then we will have the next session will be given by Ines Amin and Gongron Wan, most with a practical aspect. We will present 2 case studies using different dimensionality reduction techniques for performing multi-omics integration in the Helix pediatric exposome database, for which we will present both unsupervised model like MOFA and a supervised model like PLS or multi-block PLS in different type of health outcomes. Then we will have an important part of the course covering the topic of mediation, especially high dimensional mediation. We are in a multivariate framework, so this is not the standard mediation scenario where we have only one mediator. Here we have plenty of omic markers, and for that we will have 2 sessions, one given by Dr. Vishal Midja from Mount Sinai, and another one given by Dr. Jesse Goodrich from University of South California. And these sessions will cover, especially Dr. Vishal will cover the effect of mixtures. As we know, exposures, they have a combined effect, as a cocktail effect, and sometimes this modeling is difficult to implement it when we have multiple or high-dimensional data sets. And this will be the main focus of Dr. Vishal's talk. And then we will continue with Jesse's talk about this high-dimensional mediation with latent factors, or even the concept of gene-environmental interactions. And they have developed a very nice framework, a statistical framework to account for that, and they will present that. Then we have a couple of sessions more focused in the task of prediction. One of these objectives that Léa has presented, but we want to use the omic layer, omic markers to predict future outcomes or the risk of developing a disease. And for this, we will introduce the concept of environmental risk scores as the main tool for estimating the risk effect of environmental exposures. We will also present some very nice students from Dr. Bill Pimenov, integrating microbiota data and metabolomics data for predicting different health outcomes. And I will also give a talk mainly in trying to explain black box algorithms such as random forest or ensemble type models using the post hoc explainer SHAP. That is especially important if we want to give a proper interpretation of the most important biomarkers affecting the disease risk. And we will present a very nice case study where we have used SHAP in different ways also to identify cluster of patients with different type of risk, or even to disentangle which are the most effective preventive measures that we could do in some risk group patients to avoid the development of disease using a new technology called counterfactual examples. Then we have 2 other group of sessions that they are mainly based in the use of networks, either for visualization or for clustering. The first session will be given by Dr. Léa Maitre, and is more focused in the interpretation of findings. So once I have my output, I have identified the multi-omic signatures affecting my health outcomes. How can I interpret which biological mechanisms are behind these markers in a more holistic way? For that, we have different type of network visualization tools, and we can also do queries to different biological databases that are available with all this functional information. And we will also present again a case study published recently with Helix data in Nature Communication. And the final session is also a very, very nice work recently conducted by Dr. Nikolaos Stratakis, in which we will present another study using the method patient similarity networks to identify subtypes of disease, particularly in childhood obesity. We use this approach to leverage different multi-omics data set to identify different subgroups of metabolic disease within the complex phenotype of obesity. So in general, as you see, we cover different topics related to multi-omics integration, and also we will have an important practical dimension or aspect in our talks. We will try to give you case studies. We will try to share with you as well code for running all these methods in our GitHub project, so you can download the pipelines and apply the frameworks to your own data. And I think with that is everything that we wanted to say. Maybe now we can pass directly to, or either to questions, if there are some questions specifically about the functioning or the logistic of the course, or maybe we can just pass to the next speaker. Is there any specific question in the chat there about the?
There is no specific question. There was some doubt about the actual session dates. I invite you to check the updated PDF in the webpage, because that's where all the dates are.
Okay. So now I think we can give the floor to Dr. Arthur Tenenhaus, and Arthur will present, as I said, an overview of dimensionality reduction method for dimensionality reduction. Dr. Arthur Tenenhaus is professor at the in CERN, at the CentraleSupelec. He's PI of the Laboratoire de Signes System, and he also co-holds the APHP Andrea Chair. And his primary research interest concerns the development of statistical methods for the joint analysis of heterogeneous and complex data. So Arthur, whenever you want.
It's good. Yes, okay.
Yes.
So it's about data integration with RGCCA. So RGCCA is a very general framework for data integration. It means regularized, generalized, canonical correlation analysis. And the talk for today is about, I think, what Augusto called the vertical integration or horizontal integration. I don't remember, but the data we want to integrate is we have a set of n observation. You can see here now here we have n observation, which are characterized by different set of different sources of variable, sources of information. For instance, here you can see that one observation is characterized by J type of information, and you want to understand relationship between those J type of information. So we have J blocks of data, and we want to understand to identify for each block the set of variables that create the link between the blocks. And so RGCCA is a method dedicated to this kind of problem. Maybe I can. Okay, is it okay? You can see my screen?
Yes, we can see it perfect.
Okay, and so we want to present or to have a general framework for this kind of data integration with multi-block structure. You can see multi-block because we have several blocks of variable. And the example I used to use when I present RGCCA is an example from Russet, and it's a data set that is from Brussels from 1964, and this is the data set that are interested that want to understand relationship between political instability of a country and economic inequality. So you have two types of information here. Two types from the one end we have economic inequality, and for the other end we have political instability. And two types of information, we want to understand the relationship between these two types. And for economic inequality, in fact, we can subdivide economic inequality into two subtypes. So agriculture inequality, so the economic inequality from agriculture industry, and industrial development. So we can divide economic inequality into two types of information: agriculture inequality and industrial development. So we have three types of information, and we want to understand the relationship between those three types of information. And we have this type of information for a set of countries. Okay, you see that here we have a set of countries: Argentina, Australia, France, Yugoslavia. And for each country, we measure those type of the set of variables. So Gini, farm, and rents are the variables related to agriculture inequality. GNPR and Labo are the variables related to industrial development. And political instability, so ins x, this variable are related to political instability. And so we have classical blocks here, but that can be divided into three blocks. So the first block is related to agriculture inequality. The second block is related to industrial development, and the third one to political instability. And how to understand relationship between those different concepts. Okay, and in the multi-block literature, so what we do is to say, okay, I have three variables for agriculture inequality that are related to the same concept, the concept of agriculture inequality. So Gini, farm, and rent are related to the same thing. So they are discussing the same concept, which is agriculture inequality. GNPR and Labo, GNPR and Labo are also related to the same context that measure industrial development. And those variables are related to political instability. And you can say that you want to, dimensionality reduction is to say that I have three variables here, and I want, so these variables are maybe mainly correlated because they are discussing the same type of information. And we want to reduce those three variables to one, agriculture inequality, to find the latent factor, latent variables that reduce the dimensionality of those three variables. Okay? So I want to find a concept, latent factor, that is related, that summarized correctly, properly. Pierre and Labo, to switch from two variables to one, to switch to three variables to one. Okay, and I want to find a last latent variable that reduce and summarize those three variables. Okay, and because we want to understand the relationship between concepts. I want to understand the relationship between agriculture inequality and political instability. I want to understand the relationship between industrial development and political stability. When I do my summary, I want to take into account the link between the concept, because Rousset, when he collects the data, he was interested between some links in the data. You want to understand relationship between agriculture inequality and political instability. You want to understand relationship between industrial development and political instability. So we put link in the analysis to say that we are interested to understand this kind of relation with the data. Okay, and we are not interested to understand relationship between agriculture inequality and industrial development. So we put no links between the two. Okay, and the idea is to say, I have a kind of network of connection between latent variables, between latent factor, agriculture inequality, industrial development, and political instability. So I have network of connection, okay, designed by the expert, proposed by an expert, and taking into account the fact that we have this network of connection, I want to estimate properly the latent variable, latent factor, okay. And in the literature of dimensionality reduction or multi-block component method, usually we estimate the concept agriculture inequality by a linear combination of the variable of the block X1. So Y1, which is an estimation of the latent agriculture inequality, is estimated by linear combination of the variable of block X1. And so you can see that Y1 is estimated by a little bit of Gini, the first variable, plus a little bit of farm, plus a little bit of rent, okay? And the weights need to be estimated properly. So you can estimate them using, so for the second block, it's the same. To estimate the latent variable industrial development by Y2, you are looking for a linear combination of the variable of Y2. So a little bit of GNPR and a little bit of W21 and W22. And this is the weights that contribute to the construction of the estimation of the latent variable industrial development. And the third one is the same. We are looking for the linear combination of variable of block X3. Okay. And the goal is to estimate properly the weights. How to estimate properly the weights of the linear combination. Okay. So usually what we can do is to do principal component analysis for each block. So you can perform PC of X1, PC of X2, and PC of X3. But doing that, you don't take into account the fact that the blocks are connected, and you want to understand relationship between the blocks. So you want to take into account the connection between the blocks. And so if you do PCA, you don't take into account the connection. So maybe PCA is interesting, but it does not take into account the fact that you want to integrate all the block. Okay, so PCA is not maybe not the best option if you want to integrate modalities, because it's based on variance criteria. PCA is based on the fact that you want to maximize, you want to find W1, which contains W11, so is a vector of dimension 3. And you want to estimate W1, which maximizes the variance of the components. Okay? So in the literature, this is called block components. And in PCA, you are maximizing the variance of the block component of the score, of the lattice factor. Okay. And in the literature of multi-block, this is called the block weight vector. Okay. So you have the component here, and you have the weight vector here. And the weight vector contain the W1 contain W11, W12, W13, and you want to estimate those values. Okay, and so in the literature, so you do PCA. If you do PCA, you have a component that explains well its own block, but you don't take into account the fact that the blocks are connected. Okay, so if you want to, so we need to define an alternative, say, optimization problem or optimization criterion for estimated the block weight vector W1, W2, W3. And the two properties we want to have at the same time for the block components, so we have two properties. The first one is to say that the block components will explain their own block in terms of PCA. So we want a component like a PCA component, but we want the component to be correlated if the components are connected. You see here, the blocks are connected. So we want the associated component to be correlated. Okay. So we want a component that explains well, that is a good summary of the block. But in addition, we want a component that is correlated to its connected component. Okay. And this is the two criteria we want to impose when we will estimate the W1, W2, and W3. Block component will explain their own block in terms of like a PCA criterion. Block components are as correlated as possible for connected blocks. Okay. And in the multi-block literature, if you want a correlated component, for instance, then you have in 1961, so it's all methods, but so you can see here that all the blocks are connected. So X1, X2, X3, Xj, all the blocks are connected, and you want to find Wj, so the weight of the linear combination that maximize the correlation, all the pair of correlation between block components. So you take all the pairwise correlation between block components, and you want to find the Wj that maximize the sum of all the pairwise correlation. But if you maximize the correlation, so you force the block component to be positively correlated, and maybe you don't want to have the component to be positively correlated. That's why Kentering in 1961 and Vold in 1982 propose to maximize instead of maximizing the correlation, maximizing the square of the correlation or the absolute value of the correlation. So this is more or less the same kind of criterion. So based on correlation, and the goal is the same. You want to find the WJ that maximize a function of the correlation. But if you maximize the correlation, you lose. So you are okay to maximize to the block component are correlated as possible, but you don't take into account the fact that the block component will explain their own block. And this is this is a problem because we don't want we want stable block components that maximize that explain why it's unblocked. So you can consider an alternative criterion instead of maximizing the correlation. You can try to maximize the covariance, and I think the covariance is a good criterion because you can see that the covariance square of the covariance is equal to the variance of the block components times the square of the correlation between components of block J and component of block K, and the variance of the component of block K. So it's a compromise. If you maximize the square of the covariance, you are doing, in fact, PCA of xj through these two terms, PCA on xj, PCA on xk, but you force also the block component to be correlated. So this is a kind of compromise. When you are maximizing the covariance, you are doing a kind of compromise because you have three terms, two terms for kind of PCA, and one term that forces the component to be correlated. Okay? And so it could be interesting to have so it is interesting to maximize the covariance. Okay, and maybe we can modify a little bit this method by introducing a network of connection between blocks. You can introduce CGK. CGK is equal to 1 if block J is connected to block K, and CGK is equal to 0 if block J is not connected to block K. Okay, and so you can offer the flexibility to the user to design the network of connection between blocks by introducing the CGK. And in addition, you can maybe re-express the correlation criterion here by realize that if you force the variance of the component, so look at this. Instead of the correlation here, I maximize the covariance and with respect and subject to the constraint that the variance of the component is equal to 1. Okay, and if the variance of the component is equal to 1, this term is equal to 1, this term is equal to 1. Okay, and maximizing the covariance is equivalent to maximizing the correlation. So maximizing the covariance with respect to the block component, the variance of the block component is equal to 1, is equivalent to maximizing the correlation. And this is very interesting because we have say a single objective function, so a function of the covariance between block components, but different constraints. If the variance of the component is equal to 1, then we are maximizing the correlation. And if the variance is the norm of the block weight vector is equal to 1, then we are maximizing the covariance. So we have a single objective function, a function of the covariance, but specific constraints. If you take the variance of the component, you recover generalized the correlation criterion. And if you force the norm of the weight vector to be equal to 1, you have the covariance-based criterion. Okay, and you can from this define a single objective function, and this is what is RGCCA. It's so it's gather all this method into one single objective function. So you have a function G is a function which is equal to x square of x or absolute value of x. So G of x. So we apply a function of the covariance between block components for connected components. So we consider all the block components. We consider the covariance between block components between connected block components. Okay, and we are looking for a weight vector that maximize this function. Okay, and if and we introduce this constraints, If tau G equal to 0, then this term is cancelled. Maybe I can take if tau J equal to 0, then this term is cancelled, and we recover the variance of the component, which is equal to 1, and we recover the correlation-based criterion. If tau J equal to 1, then this term is cancelled, and we recover covariance-based criterion. And when tau J is between 0 and 1, then we have something between correlation and covariance, and it plays it is a kind of regularized version of canonical correlation analysis. So tau J plays the role of regularization, which is useful in a high-dimensional context, which is the case in multi-omics data. So RGCCA is defined by this optimization problem. It's a very general framework for multi-block data analysis. And we can discuss maybe the two-block situation when you have only two blocks. So in the two-block situation, this optimization problem reduced to this one. So we are maximizing the covariance between block component of block X1 and block component of block X2, subject to these constraints. And it is quite interesting to realize that when tau J equal to 1, when tau 1 equal to 1, and tau 2 equal to 1, then we are maximizing the covariance, and we recover Pell's regression, which is a well-known method in the data reduction in the multi-block literature, two-block literature. When tau J equal to 0, for tau 1 equal to 0 and tau 2 equal to 0, then we are maximizing then the variance of the component is equal to 1, and we are maximizing the correlation. Okay, and this is exactly canonical correlation analysis. Okay, this is also one method in the multi-block literature, and multivariate data analysis literature. And when tau 1 equal to 0 and tau 2 equal to 1, then tau 1 equal to 1, sorry, and tau 2 equal to 0, then the criteria becomes this one. And this is redundancy analysis, also called reduced rank regression. And you see that in that case, the optimization problem is not completely symmetric here and here. It's completely symmetric. Here you are maximizing the covariance between X1 and W1 and X2W2. It's the same as maximizing the covariance between X2W2 and X1W1. So this criteria is completely symmetric. This one is also completely symmetric, but this one is not symmetric. And you put in the last one, so in this optimization problem, you force the block component associated to X1 to be like you see that the variance of the component is present in the criteria. So you are doing a kind of PCA criterion. But what you are doing also is you force the component associated to X1 to be correlated to the component associated to X2, okay? And you don't want a block component associated with X2, which is correlated, which will explain its own block. You can see here like this. Component X1W1 and X2W2 are well correlated because of this term in the optimization. You have a stable component for X1 because the variance of the criterion is maximized, okay? And there is no stability condition for X2 because you are not maximizing the variance of the component for X2. Okay. And so you can see that if you want to choose a good value for tau G, if you choose tau G equal to 1, you are close to 1, you favoring stability. And if you choose tau G close to 0, you favoring correlation between blocks, between modalities.